A very good evening to one and all present here. So today the lecture will be delivered by our Honorable Director, Dr. GN Sastry. So now I request our director to deliver his lecture.
So as most of you know, this drug development is a process which is it audible? So the drug development is a process in which you make a molecule or a compound which will be affecting as a drug. So let us see the journey that a molecule takes when it is orally administered. There are different types of administration, and once a molecule is orally administered, it has to dissolve because the water is a medium, and it has to survive a range of pHs. It has to survive a range of pHs, and it has to go through the gastrointestinal tract where the pH range of 1.5 to 8.0. And more importantly, it has to survive intestinal bacteria, and it has to cross membranes, and it has to survive liver metabolism, and also it has to survive what is known as first pass mechanism, avoid and avoid excretion by kidneys, and avoid partition into undesired places. Like, for example, a molecule is there which has a target in a particular organ. It should not be targeted to central nervous system, so the blood-brain barrier should be sufficiently high. So if there is a molecule which is sort of taken by the pregnant woman, it should not be affecting the fetus unless it is designed to do that particular job. So all of you have a very simple understanding that this drug discovery can be reduced to lock and key type of a concept. That you have a key which is a small molecule and a lock which is the large molecule which is the receptor. And in a very, very simplistic sense, it can be reduced to the best fit between the lock and the key. And you have to find out the right key for a given lock and so on and so forth. However, it's a little bit more complex than what one thinks. And a particular molecule should not only fit as a lock and key, it should also have selectivity. What does it mean? It should do a particular job, but it should not be doing jobs which are not assigned to it. Because usually the molecules may be efficient and it might have a problem of toxicity. Toxicity, one way it arises is because the particular small molecule will be interacting with receptors which it should not be interacting. The next is reaching the target or the bioavailability, which depends on a large number of factors like pharmacokinetics, pharmacodynamics. And it's very important that the particular molecule should reach the target. And more importantly, one should be able to synthesize this molecule or get it easily from a natural source. And economics play definitely an important role. And more importantly, people can develop a molecule. It might be a wonder molecule and it can be working, but after some time, the molecule will not be effective due to the development of resistance. As most of you know, probably because of current situation COVID, the viruses, if there is a viral target, that gets mutated. But sometimes even when you don't have a viral target, even if you have a host targets, they will also develop resistance, the small molecule. And there are several reasons why it develops resistance. And one of the reasons is the efflux proteins will get activated, and like P-glycoprotein and other efflux proteins, which will make the molecule not available. The bioavailability of the molecule will be reduced because these particular proteins, when they are overexpressed, they will be throwing the molecule out of the bloodstream. And sometimes the chemical stability is very important. There are a large number of molecules which are used as prodrugs. Prodrug is the one wherein you take a molecule and this particular molecule will release the active form, and that will be efficient. But sometimes even this molecule or an active form, their stability play a very important role, and many times it might be depending on the individual. So pharmacokinetics is also very, very important. Pharmacodynamics is very important, and pharmacogenomics also is a very, very important factor for a particular molecule to be a drug. So many times a drug need not be the most potent, because sometimes a less potent one, a drug which is less sufficient may work because you will be able to adjust the dosage to have the maximum, the optimal effect. And a drug need not be the most stable. Although stability is a, the potency is a very important criteria, but the most potent molecule need not be the drug. And similarly, stability is a very, very important criteria. And the most stable molecule need not be the drug. Absorption is an extremely important property, but the molecule which absorbs the best need not be the best drug molecule, nor the least active against some other targets which are not desirable also is not effective. So, for a molecule to be a drug, it has to satisfy a number of criteria, and it's a multifaceted thing wherein one will have to optimize all these conditions, and the best balance will make a molecule the drug.
No, now he is talking. He is not showing any slide.
Hello. Is it? It is often very important to understand that there are a large number of factors which might work in concordance or discordance, which will make a molecule a drug, and it's extremely important for one to realize that one has to balance all these factors. So let me try to slowly introduce you to this drug discovery as one phase and drug development as another. So in general, people have to understand that a typical process to have a drug from the beginning will take about 10 to 12 years, and the average cost for such a event to take place is about 1.5 to 2 billion dollars, which is a huge amount. And you know that $1 billion is about 100 crores in Indian context. $100 crore is a lot of big budget, and a large part of this money will be used in the development, that is clinical pipeline. So there is a discovery phase, that is preclinical pipeline, which may take about 3 to 4 years minimum. So if one starts from the scratch, one has to find out what is the potential target, validate it, and get what is known as high throughput screening, and then they get hit molecules which will show some efficiency against a particular target, and then one has to get a lead molecule that is hit to lead, and then the lead need to be optimized, and one has to go through a whole process of in vitro and in vivo processes and animal models, and come to a stage where they will complete preclinical development and go to the next phase, which is called development. The preclinical pipeline till now is known as discovery. Discovery is very different from development. And initially, I thought that I will give one lecture on discovery, one on development, and third on computer-aided drug discovery and development. But however, today I thought that I may give a little bit of introduction of what is the drug and then talk about development, because discovery is a lot more scientific, and I will try to talk about drug discovery tomorrow. So when you go from preclinical pipeline to clinical pipeline, you have phase 1, phase 2, phase 3 clinical trials, and this may take about 8 to 10 years. It is probably one of the most expensive. And today, I will try to throw some light on what is a phase 1, phase 2, phase 3 clinical trial, and how this is very important to understand. And then, after the clinical trials are finished, one gets FDA approval. So this is a discovery phase is up to preclinical, and development phase of a drug is the most important. So there are several factors, and then people can worry about many factors. And after doing preclinical trials, they will try to go to drug development. So today I will spend a little bit of time on drug development and drug discovery. So a drug is a novel molecule, and there are many factors which are involved with a drug. Intellectual property is very important. For example, if someone discovers that this is a drug molecule, who will own the property? It is very important, and one can know the structural activity, the pharmacokinetics, pharmacodynamics, safety, and many other aspects. So one has to realize that there are different types of people who need to be involved to get this understanding, like biochemists, pharmacologists, immunologists, DMPK, enzymology, and there are different types of people like synthetic organic chemistry, physical chemistry, combinatorial chemistry, and there are several other factors which are related, and computer aided drug design and modeling also play a very important role, and currently information technology is playing an extremely important role. So essentially you design a novel molecule and you get access to this, and then you have access to this novel molecule, and afterwards you have to protect it by writing the patents, and then one has to understand all the data that is there, and then look at the safety before they can go to development stage. And there are some major challenges in lead optimization, like one is the bioavailability and the systemic exposure. So how the particular molecule is distributed inside the body, and it's also important to see what is the clearance, and the first pass clearance is important, and absorption is important. Gut stability is also very important, and the clearance through renal, plasma, liver, all these things are very important. So afterwards, you have the metabolism, cytochrome P450 type of enzymes, which are usually very, very specific to different individuals, wherein pharmacogenomics play a very important role. What does it mean? There is a molecule which is administered to one individual, it behaves in one way, and if it is administered to another individual, it will behave in a slightly different way. So basically, one also have to understand that how different people behave in different ways. So there is a lot of science in it, and one has to understand that drug discovery is an extremely challenging in its own right. And it is essentially science driven. And once somebody completes that, they will have to go to the development, which is a high budget affair. One has to go from clinical to preclinical to clinical, and the first step is they have to file what is known as investigational new drug applications. I will not use a lot of animation. So when they have the preclinical data, they have to look at the safety assessment, ADME properties, look at the pharmacology, process R&D, pharmaceutical R&D, bioprocess, clinical investigation of the patient, clinical pharmacology, regulatory, and statistics and epidemiology is very important. And you will understand that even in the COVID situation, it's extremely important to know the value of data analysis, statistics, and other data-driven techniques like data coordination, information services, research information, and epidemiology. And then after getting this information, they will formally start the clinical trials. In the clinical trials, for any company to start the clinical trials, they will have to get the clearance from FDA for the IND, investigational new drug application has to be filed with all the data, and FDA will give the approval. I am talking about the US FDA. And in India, also we have our own regulatory, but this is something which is the international standard, and because of some of the agreements that we have, we will follow. So, in these clinical trials, you have phase 1, phase 2, phase 3, and also phase 4. In the phase 1, you take a small number of healthy volunteers, that's ranging from 20 to 100, and you give this drug for about 1 month. This is all what is done. And what do you get? You get a product profile, and you also have the information on the absorption and metabolism, and what is the effect on organ and tissues, that is the toxicity on real individuals, and the side effects when the dosage is increased. So this is an extremely important step, that when somebody has a drug, the basic condition in phase 1 clinical trial is it should not be having an adverse effect on a healthy individual. The question is, which healthy individual will agree to get it tested? So there are different protocols that are there. There are biosafety, bioethical committees, and ethical committee play a very important role. And it is not possible to administer these types of new molecules into anybody, and it is very strongly controlled by the regulatory, and one will get an information whether the molecule which was attempted to become a drug will be safe or not safe in phase 1 clinical trial. And when you go to phase 2, we will have a very extensive study about the efficacy of this particular molecule in the health impaired patients. So you have to have a control group which consists of a number of health impaired patients, and to validate that this particular molecule or a drug is working or not, you need to have a treatment group. So you have a reference group or a treatment group, and you also have a, sorry, the treatment group will consist of health impaired patients, and the control group will be a reference group, which is a healthy volunteers. Again, the information that you will have is effectiveness in treating diseases and short-term side effects, and what is the dose range will be obtained by phase 2 clinical. Then the third clinical trials will be there, which will have an exhaustive testing of the dosage and dose optimization by taking hundreds and thousands of health impaired patients. So after finding out that after a particular molecule clears the phase 2, it shows efficacy because with respect to control group, the treatment group will show much better effect. Then you will have the phase 3 clinical trials, which will give robust information about the benefit versus risk and what are the short-term and long-term side effects. So necessarily, this will be taking a lot more time compared to phase 1 and phase 2. So at this particular point of time, people in principle can go for a very, very limited compassionate use. And the clinical trials will be continues to be conducted, and this data, the whole data, will be given to the regulatory agencies. In this case, as we discussed, the Food and Drug Administration, FDA. So worldwide marketing authorization will also be given. So one will apply, the IND will be given a status of NDA. New drug application will be given for FDA, and it will have to go through the entire clinical trial data will be submitted to the advisory committee. There are regulatory review teams, and they will give their reviews and comments and discussions based on every aspect of that particular molecule. And the drug companies and regulatory liaison activities will be there. And finally, the FDA will give approval for the new drug. And even when they give an approval for a new drug, it will go through what is known as the post-market surveillance, or after FDA approval also. One will be closely watching whether there are any side effects. And a number of molecules were withdrawn even after they get the FDA approval. So, if you see the process, one has to start with a large number of molecules, maybe several lakhs of molecules, which will pass through all the preclinical data, and about 100 molecules, if you send it to the clinical trials, at the end of 10 years, about 2-3 molecules will come as drugs. So it's a very expensive and very risky business, and then a large number of such molecules are discovered actually in some small laboratories initially, but they don't have the capacity to pay for all the clinical trials. So big pharma giants will buy this knowledge, because if there is a molecule which will show a lot of activity, if you want to make these clinical trials, it may cost a lot of money. A typical clinical trial at least will be about a few hundred crores in India to complete. And at the end of that, if it fails in phase 1 or phase 2, all this effort is gone. And you have to start from square one. So a large number of small companies, if they put all their efforts into one molecule, and if that molecule fails, there is no way that the company gets recovered. So what actually happens is, in a number of cases, the small molecules, when they see that there is a potential to sell their knowledge, their data to a bigger company, it will be good for them because they don't have to take too much risk. So invariably, in drug discovery, the large company will get a chance to have a blockbuster drug. And even with the best of the best minds and with the best of the best interests, it will be very difficult for a small company to have the whole drug discovery, cross the whole drug discovery pipeline and also development pipeline, and then come to a stage where they get the FDA approval. From then onwards, they can sell and get some profit if they get the approval. So how long is the wait? A minimum of 12 years and maybe 15 years or maybe more. So this is a very, very complicated circle. The drug development circle, as somebody has put, the research component in the x-axis, you have the time, and from extreme left to the bottom of the curve, it will be around 12 to 14, 15 years. And you will have a research and development. And after the research, after the development, the technology will be transferred. Still, you have to invest money. And then there will be a product launch. Still, one has to invest money. And one has to invest money even after the launch of the product during the commercialization. It may be a few years. Because just because somebody got an FDA approved drug, people will not be buying it and then using it. So there is a lot of requirement to make it commercially successful. Maybe there is a drug which is having an FDA approval, but there may be another drug which is better than this. So people will not buy. So commercialization is very important. So what exactly happens is people keep on spending money and money and money even after 15 years or 20 years or so. They don't see the curve raising. From the bottom, you see that this curve is rising, and then that is not happening because it will be taking a lot of time and the company should have a lot of interest and then resources to sustain. So this famous thing is known as Valley of Death. A large number of pharmacy companies will be meeting the Valley of Death if they wanted to really take a molecule from the beginning till the end. So this is very, very important for people to realize. So many times at some stage in research or development, they will sell it to a pharma giant and then they can take a risk. If they are failing even in 3-4 cases, they may be successful in one case and that will take care of the company for several years. So planning and focus on clinically relevant areas, and it's extremely important to wait for a long time. So once the product is commercially successful, it will take some time to recover all the investment that was made from the beginning in this particular drug development. And only after this point is reached, which is a very, very commendable feat, only a few people could achieve that. From then onwards, whatever you get is profits. So if there is a blockbuster drug, that will give you a lot of strength to do research in areas which are not touched upon, because you will get so much of money or a profit if one of the drug will become a blockbuster. So I always feel that our educational system is so much into teaching people on the importance of doing things correctly. This is a rule, and you have to do this in a correct way. You have to take an exam, and you have to get highest possible marks. Or you participate in a electrician competition, and you should be the winner. Or you write an essay writing competition, you have to be the winner. So this particular rigorous training to do things in a very, very correct way and in a chosen way is really good. But I think what is more important is doing correct. You may not be able to do things in a perfect way, but sometimes you have chosen some work which may not be very relevant. Then doing it perfectly or not so perfectly may not make a big difference. So what makes a real big difference is one has to do correct things. So with this, I just wanted to tell you that drug discovery and development is a very challenging, very, very challenging issue. And then many of you should understand that although people say that there is a vaccine or there's a drug, vaccine is also very, very similar to this. And I feel that although large number of people say that one may get a vaccine very quickly, the safety will be an issue. So then you can say that can we not get a drug in 10 years? No, it is not like that. In cases which are very important, for example, when HIV has become a big and dreaded disease, and people have got the HIV drug in the record time because they are able to relax a large number of relax a large number of rules and then did not wait for the complete trials, and they when they are satisfied they have recommended, and then people can get it even as quick as 8 months if a particular candidate is ready. But however, one has to realize that if a particular molecule will have a side effect after 1 year, which is the case, there are several examples. One of the most notorious example is thalidomide. Thalidomide is a drug which was used for a morning sickness. A large number of people have used. A large number of people have used, and all of them, or most of them, has a huge problem. Most of them have a huge problem with the children that were born for the person who has taken this therapy. So I feel one has to understand the enormity of the situation. When something is very complex in the general context, one will say that this is like a rocket science. Why? Because you put rockets, and this rocket, if they want to have a successful attempt, it has to exactly go to the orbit and it has to meet all the conditions. The escape velocity, what is the load of it, what it carries, all of them are very, very important. So, if someone wants to achieve a perfection, it should be like rocket science. I think when a rocket fails, you will know that there may be this reason for which it failed. But drug discovery is often even more complicated than rocket science. One has to understand why. Because a particular molecule might fail for various reasons. One example may be, after all these attempts, one can make a molecule, but the disease may not be there. There are many diseases which are eradicated. That's a good way. The company will not make a profit. But there may be, after all these efforts, you develop a drug, and there may be another drug which may be more efficient and cheaper, and so on and so forth. There are a large number of cases where the drug discovery is a phase of great interest, and drug development is a completely different ballgame. And I hope that many of you realize the difference between drug discovery and development, and try to comprehend the enormity of the situation and see how difficult for small companies to go for drug discovery and development. So even for countries like India, it's a daunting task. So there are different things like one can collaborate, or you have approaches like drug repurposing. And drug repurposing is the one wherein there is a molecule which has already got an FDA approval, but this FDA approval was obtained for against a particular disease. Now you can see whether this particular molecule will be efficient for a new disease. For example, if it is COVID, you can have an antiviral molecule and then see if this antiviral molecule can also be effective against COVID. So that is the reason why drug repurposing has become a very, very important tool in this area of. And if you want to develop vaccine and other things, we can try to look into the vaccine design. Somebody will give a talk later. And tomorrow I am going to give you a talk about drug discovery process. What is the hit, lead optimization, and what are the steps that are involved, and what are in vivo and in vitro methods, and what are animal models, and how do we develop. And day after tomorrow, I will try to talk to you about computer-aided drug design, computational approaches in the drug discovery. So today, I think I have given you an overview about what is a drug and how it acts, and then try to talk to you about the economics that are involved and the drug development process. So this is very, very important for people to realize, and I will be very happy if some of you can get some interest in this and then find out that how India can be agile. Because many of you may be confused sometimes. People will tell you that a large number of molecules that are made in the world are made in India. Large number of tablets. That is true. And these are all in a particular area which are called generic drugs. What is a generic drug? I told you the drug discovery and development process. After you develop a drug, at that particular point of time, the company would have spent a lot of money on that drug, and it would like to get the profit. So when it wants to get the profit, the knowledge has to be protected, and no other company can make these molecules unless this particular company who is the owner of the intellectual property will allow the other companies to synthesize. Okay, and that particular right will exist with a company for 20 years. And after 20 years, the molecules which will go off patent, the patent protection will not be there. So after 20 years, if there are some companies which would like to synthesize this molecule and then sell, they can sell it. And these are known as, the molecules which are off patent are known as generics. So when you go to generics, you know that this is a molecule which is efficient and a particular company probably is selling for a high price, and then you can try to decrease the margin, sell for a low price. Or more importantly, you can come up with a new synthetic route and then try to decrease the price. This is one of the greatest strengths that India has. Indians have shown in a number of cases that they have reduced the cost of these generic drugs by an order of magnitude. In this, I would like to particularly bring to your attention about HIV drug. The HIV drug treatment it used to cost about $1,000 per month for an individual. And large number of people in Africa got affected, and there is a huge issue of how we can spend so much money on an individual patient. So, CSIR, IICT, and NCL, wherein Dr. A.V. Ramarao was working, along with CIPLA, they have reduced the cost of production of these HIV drugs from a large amount to less than $1. So it is a 1,000-fold decrease, which made the drug available to a large number of people. How did they do it? They tried to develop a completely alternative process, which is very, very economical to produce this drug. So science play a very important role. Synthetic organic chemistry will play an important role in the generic drugs. But if you want to get the new chemical entities, it is a very, very challenging problem. And I would like to tell you that about 50 years before, this is under the domain of pharmacy people and natural products extraction. And then people find out that how to get a molecule from the natural products. But after late 70s and 80s, the medicinal chemists played a very important role in drug discovery, and synthetic abilities of the medicinal chemists have become very, very high. And slowly, in 2000 onwards, biologists play a very important role. And when it was with natural products chemists, the drug discovery and development prices or costs are very low. And when it is in the hands of chemists, the price, medicinal chemists, the price has increased. But when it is in the hands of biologists, the cost of production of drugs have skyrocketed. And in recent years, in addition to the huge interdisciplinary team, people have realized that artificial intelligence and machine learning coupled with molecular modeling play a very, very important role in drug discovery. This is something that our group is trying to work. And tomorrow, I will try to talk to you about drug discovery and what are the methods. And day after tomorrow, I will try to talk to you about computational methods and also the efforts that we are doing in our group in this studies. So I see that there are questions, and I'm very sorry for this late evening timing, because I feel that this is probably the only way that I can be a little bit more sure that I can deliver a lecture. So one question I think with this I will close my talk. And there were several questions, and then probably what I do is I will try to answer a few questions now for the next 10-15 minutes. And if there are these questions, I will try to answer them offline. I see Ambarish Raj Teka says that my question is that graphene is nowadays used in drug delivery and cancer therapy. Can you please throw some light regarding this topic? This is a very interesting question, and it's very important that drug delivery is very, very important. And there are several carriers for drug delivery, and graphene is one of them. Graphene is something which can be functionalized in various ways, and the functionalized graphene has very, very important properties, especially in drug delivery. And depending on the customized functionalization, it will be possible for the molecule to be delivered at the right site. This is very important for the bioavailability. And many times, the stability of the molecule is a problem. So, if a delivery carrier of that molecule is there, it can encapsulate it and then prevent it from dissociating or becoming unstable in various media. Only when it reaches the target, it will release it. Or sometimes the molecule will have difficulty in permeating through various phases, and in that cases also molecules like graphene or something cationic lipids and other molecules can be used as drug delivery agents, and graphene has proved to be fairly effective. And of course, we will also have to look at the toxicity and other. What is bioavailability and bioequivalence? Bioavailability is the availability of a molecule. Once it is administered, the molecule should be reaching the target. Bioequivalence is mostly in the pharmaceutical company where the dosage, once it is there, it has to be maintained. It is something like a quality control type stuff, right? And all of you should realize that a particular molecule, when it is administered, it will enter into the blood, and there is a therapeutic window, and if the molecule's concentration is within that therapeutic window, it will be efficient. It's working fine. And if the concentration is a bit too high, it will be toxic. If the concentration is lower, it will be not efficient. So it's very important to be able to get that particular equivalence with respect to a standard drug. So if some other company is making a particular drug, and if they are not able to produce the active ingredient of that particular molecule, then the bioequivalence is not right. So because there are many cases where people use excipients in your formulations, and depending on that, the bioavailability will be very. So someone is asking, can you please give us a brief idea on open drug discovery tools? And this is an important question, and then I will touch upon it day after tomorrow. In our group, we are developing a software which is called Molecular Property Diagnostic Suite, wherein we are trying to get most of the open source drug discovery tools, and they will be available. These are the tools which are available for people to use, and one need not buy. One may download it, or one may write to the developer and they will send a copy and one can use it. These are called open drug discovery tools, open source. So there are different questions which are related to project. So there is a question, can CRISPR play any role in developing drug against corona? Is any experiment going on in this on the same? This is a very interesting area, and then people are working in this, not only abroad, even in India. Some groups are working in the CRISPR-Cas based technologies. So another person asks, as viral targets tend to mutate, so what should be the drug discovery and drug development plan against viral targets, specifically in case of coronavirus? So this is a very interesting question. That's the reason why even for a common flu or influenza, one is not able to develop a vaccine or a drug. But what was it's too early to say. The mutation rate of this coronavirus is not too high to preclude the drug, and hopefully drugs can be developed. As far as we know, the mutation is not as fast as in other viruses, but this has to be verified, and then people are now only accumulating the data. But this is a very important question, and then one has to consider this aspect. The mutation in viruses is really important. Like even bacteria also, people should try to look for alternatives or multi-drug therapy. For example, in a particular place, it gets mutated, you can try to look into an alternative active site, and there are different ways in which one can deal. The drug discovery is made only in dry lab conditions. No, drug discovery is not a computational exercise. It is a bit of computation, but largely experiment. So definitely drug discovery has to be done in the laboratory, which involve medicinal chemists, biologists, pharmacologists, and many animal and experimental trials. That is the discovery process. Before they go to development, drug discovery has a huge experimental component. But if you can judiciously use computation, it will be very beneficial, and that's what people are doing. Does the molecular mass play any role for a molecule to be a drug? This is a fantastic question. Molecular mass play a very important role, and molecular weight, right? This is called, and this is one of the important determining factors in the Lipinski's rule. And as the molecular weight increases, the drug-like properties, it is very difficult to optimize. So it's better to have small molecules with high efficacy as a hit or a lead, and it should not go beyond a particular value for a molecule to be effective as a drug. But there are also biologics, that's a completely different story. How long will it usually take for a drug to be discovered and marketed? This is what I have been telling, that it may take any time between 10 to 15 years. But in some very rare cases, one can get it in a much less time. Like, for example, if you want to really press all the regulatory things, one may get it within a year or so. So in conditions like COVID, it will be possible that one can take a drug, but this is a very complicated thing, because you will have to use some shortcuts, and safety has to be considered. And then maybe this can be given to some healthy individuals, but if you can give to the people who may be vulnerable for a particular drug, one has to be very careful. So it depends. It takes a minimum of a certain amount of time for the clinical trials to be done, at least an year or so. But on average, it's about 10 to 12 years. I want to know whether there is any natural resources available to combat the resurgence of corona. Large number of natural resources are available, and one has to be tested. The only thing is validation is important, and then we have to know whether even these natural products also might have some side effects, and then one has to look into it. So I have a large number of questions, and some of them are some question which is a little bit interesting. Taking more dose of a drug, is it the reason for development of resistance? More dose of a drug will result in toxicity. Usually, for developing a resistance for non-viral type of drugs, it is to do something with the expression of the genes, or something should happen in the body, right? So it is usually associated with prolonged and chronic use, not necessarily due to the excess dose of a drug. It is due to the chronic use. For example, chloroquine is probably one of the most important drugs, and it's also very cheap against malaria. But this Plasmodium falciparum, this malarial, will develop resistance for that, and then one has to go for an alternative routes, like that antimycin or some other treatment, and after some time the resistance will stop, and then even chloroquine will start working. So, can we expect the discovery? If the clinical trials takes years' time, can we expect the discovery for COVID-19 by November? This is a very, very relevant question, and I know that many people will be wondering about it. Definitely, it's possible. If somebody has a lot of confidence that this might work, we might get a drug approval at a very, very fast rate, especially when the governments are proactive and the regulatory agencies are sort of convinced with the type of data that is produced. So I think to stretch this lecture beyond 1 hour, I would rather sort of close this. Is it possible to have all these questions recorded? Yeah. So my team will record all the questions that are given, because there are a large number of questions, more than 250. So many of them are very, very interesting questions, and we would definitely like to answer them. And I will try to give answers in our website or a blog or whatever is possible in the next 1 day. So with this, I will be closing this talk, and I hope that please explain valley of death. I will explain this valley of death, as I explained that the cost of these drugs are so expensive, and many times one wonder whether they will get the market, and a process which has started to make a molecule as a drug will not yield anything. You spend a lot of money, several crores of money, crores and crores of money, and several years of your effort will not lead to anything. That is known as the valley of death. Okay. Because you're not going to get any profit. So many interesting questions. I'm very happy. Now the questions are increasing even now. So in principle, I can go for some more time, but to make it not very, very long, with your permission, I would like to stop it now. And then I will see you tomorrow evening at the same time at 9 o'clock. And then we will have probably some of these questions, if they're interesting, I will answer during my lecture. And then I will try to give you a description about drug discovery process in a little bit more detail than what I have done today. Thank you very much. Good night. And I'm very, very happy to see that more than 400 people have attended, and some of them might have been interested in this talk, and I wish you have some good experience because of SRTP. Our team has been putting a lot of effort. In Assam, we have not only COVID, but floods and many other things. And despite that, our team is working very hard, and hope that you will be able to enjoy some of these lectures. And many, many thanks for your patronage. And then keep attending the lectures, and then try to do something either with the mentors or with others, and then keep the scientific spirit high, and let us have some fun doing science in the next 45 days or so. Thank you very much. Good night and Jai Hind.